Ah, merci. Merci tout le monde. Merci Pauline pour l'invitation et l'opportunité d'essayer un peu de français. Je suis contente avec ça. Yeah, so thank you so much everyone for coming. I know we have quite a mixed audience, cognitive neuroscientists, clinicians, engineers. So I hope that the title is a bit long and involved, but I hope there's a little bit of something for everybody here. Maybe not going into too much detail in any one thing, but happy to talk after about anything that might interest you. So the format of the presentation is actually this was based on a presentation that I gave in an interview for a grant for the Wellcome Trust, which is one of the largest funding agencies in the UK. And I got the grant, so it's kind of explaining what I'll be doing during the grant with a bit of background. So it's a bit more my aims for the project with a little bit of science. Yeah. So my work is studying depression and trying to understand depression. It's one of the largest public health burdens in psychiatry and in the world, has a huge economic cost. It increased quite substantially during the pandemic. It's gone down since, but it's still a huge burden. There's an interesting statistic. It costs mental health disorders and depression cost the UK about £270 million per year, and the estimated cost of the pandemic was about £300 million per the year. So it's the cost of mental health issues on society is something like having a pandemic every year. That's the type of burden that we're dealing with. So one of the main symptoms of depression is anhedonia, which is a lack of interest or pleasure in activities. It can be quite debilitating. It can reduce motivation to work, to see friends, and to do the daily things that you need to do. So, but anhedonia is quite an umbrella term. It means quite a few different things depending on how you characterize it. Lack of volition, interest, pleasure, motivation, vigor, drive, all these different things that can go into this one clinical construct that we use. One of the most profound ways that people have deconstructed anhedonia is into these two subdomains. So anticipatory anhedonia, so your motivational drive or how much you anticipate pleasure from an activity or your need to do an activity. And then consumatory anhedonia. So when you actually do something, how much pleasure and enjoyment do you get from it? So a bit more of a hedonic process. So they're quite useful ways of deconstructing anhedonia. And this is important because we need to know in our patients and our participants in ourselves, what is the problem? What is the thing that we're dealing with? What do we need to tackle? Are there different subgroups of patients that we can deconstruct and then treat them more directly in a more personalized manner? So it's important to try and deconstruct these broad symptom domains. It's not just relevant for depression. Anhedonia is relevant for a range of psychiatric and neurological disorders. It might be called apathy in Parkinson's, and we're often talking about similar overlapping constructs. So if we can understand these types of behaviors, it might have far-reaching implications, not just for depression, but for a range of disorders. So yeah, I think it's critical to pause. It's quite severe, just to say as well, in terms of severity. People with depression who present with anhedonia, they tend to have poorer outcomes and they're at higher risk for treatment resistance. So it is quite a severe symptom to have. It's multifaceted, as I mentioned, and transdiagnostic, so it's relevant to a range of disorders. So it's important to try and understand it, measure it, characterize it. And so how do we usually measure anhedonia or the behaviors that contribute to anhedonia? We use these cognitive tasks, usually effort-based decision-making tasks for money. So you present someone with different levels of money, you ask them to do some work for the rewards, and you see how much work they'll put in for various amounts of rewards. That gives us some kind of measure of motivation. And that's one of the things that we can use to measure anhedonia in a more objective way. And we know from a lot of preclinical literature and work in rodents that the dopamine system is important for modulating this kind of effort-based decision-making and reward valuation. But we know it's not just money that drives behavior. There are these so-called intrinsic factors as well, things like achievement and autonomy and enjoyment of an activity just for doing it in and of itself without any extrinsic reward. So in psychiatry and neuroscience, by just using money and understanding how people put in effort for money, we're kind of ignoring a whole range of behaviors that are a bit more ecologically important, in terms of people just doing things for the sake of it, or for some sense of control in their environment, some mastery, and some sense of achievement, like I've accomplished something, or I've gone out today, I've had a shower, that kind of thing. We need to, that's nothing to do with money and them gaining money. So yeah, we need to kind of explore these factors as well. So that's sort of the first aim of my project, is to try and characterize these sort of intrinsic and intrinsic factors that might drive behavior. So the way we plan to do this, and we've started collecting a bit of pilot data, but not much. So we have this task. So people see these different screens, and they have to move this avatar down a path from the beginning to the end of the path with some button presses. And along the way, in different quests, there might be different reinforcers. So in one context, at the end of the path, there's some money, a pot of gold. In another one, can you see my mouse? See the mouse? That. So a pot of money, that might represent an extrinsic reinforcer, to see how people respond to that. In another one, there might be a small challenge that they have to overcome, for example, passing these barriers. And if they're able to pass them and get to the end, that's what we model as a sense of achievement. And then in some conditions, there's this brain slug that will come on the avatar. And so that diminishes the sense of agency, because their button responses are actually divorced from how the avatar moves. So they're losing a bit of control in terms of that quest. So in this sense, this is how we're able to model the different effects of things like money as an extrinsic reinforcer, a sense of achievement, and autonomy as these sort of intrinsic reinforcers. And then in some different, we have a few different versions of the task, but in one of them, we simply ask participants to bid to go on each of these quests. So they learn about the quests, they know how they feel, how to experience them. And then we tell them there'll be a forced play period where they have to spend about 10 minutes doing these quests. So right now you should bid on the ones that you want to go on. Where bid represents their motivational value for each of these different factors. So then we try to predict bids in a very small sort of pilot sample, and we've replicated this a few times now, but just to show you, so this is how much each of those different factors predict their bids or predict their motivational value. So you see a sense of autonomy positively predicts like how willing they are to go on these quests. That the money is quite strong and significant. It's usually quite a driving factor. These are healthy controls, so it's usually quite a driving factor. And there is a small contribution to this sort of a sense of achievement. And I think this is interesting because this is how much they want to go on this quest with the barriers versus a quest with no barriers. So there we're really talking about them wanting to overcome some challenge and getting to the end, even though the probability of getting to the end is the same in both situations. It's this added challenge that they're able to overcome. So that's how we're able to model sort of a sense of achievement. And what we saw in a very small sample of people with depression was you really saw the difference in that sort of achievement motivation context. You saw a slight reduction in the money, in motivation for the money trials, but you really did, we did see quite a big reduction in their motivation for that achievement, overcoming a challenge trial. So this is one of the ways that we think we're going to be modeling this kind of intrinsic motivation versus motivation for extrinsic factors. And going back to how that relates to symptoms, because it is really important to try and deconstruct symptom severity and these different constructs. I'm not plotting the data, but I can show you later. But we did see a relationship between this sort of achievement motivation, this intrinsic motivation, with anticipatory pleasure or anticipatory anhedonia. So the more motivated you were for these intrinsic goals, the more you were able to look forward to something, and you had some more prospective interest in activities in the environment. Whereas there was a relationship between your extrinsic motivation, or how much you wanted the money, and consumatory anhedonia. So the more you sort of wanted the money and these extrinsic factors, the more pleasure you got from those kinds of outcomes. Okay, so that's sort of a very brief overview of the aim one and how we're going to measure different types of motivation. So the aim too is all about using high field MRI, and I used to call it ultra high field, but now I'm here. I definitely can't call it ultra high field. Absolutely not. So it's using 7 Tesla MRI to study, the dopamine network. So as I mentioned, we know that this system is important for modulating these kinds of drives and reinforcement. And so I like to, this diagram is, so the dopamine system stems largely from small nuclei in the midbrain, namely the ventral tegmental area in red there, the VTA, as well as the substantia nigra in green. And they project throughout the cortex and to the limbic system, subcortex. This diagram is very generous to the VTA. It's not that big. So this is just a T1 image where I just drew a little dot where the VTA sort of actually is in its approximate size. It's really, really tiny. And so when I started my postdoc and I had some functional images, this was the type of data that I was dealing with. And I know we can do so much better now with 3T, but I like to show this just to show the limited resolution we have to measure these very important small nuclei that are so important for these psychiatric disorders. And of course, as I think most of you know, with higher fields, we can get higher precision, spatial resolution. So this is an example of a functional scan at 7T that we acquired at Mount Sinai. So it gives us much more detail and precision to be able to discern these signals from these small regions of interest. And not just, we don't just get gains in terms of TSNR, we get these really much improved spatial maps of connectivity estimates, for example, which is what this is showing. I think I'm probably preaching to the choir. So just to give you an idea of some of the tools that I'll be using during this project, and I have used in the past to study the dopamine network. So I'll be using so-called neuromelanin MRI, but it's not. But so this is using very high resolution structural MRI. So it's about 400 micron isotropic structural slab through the midbrain with 7T. So it's so magnetization transfer contrast. So that gives us a proxy measure of large macromolecules as well as neuromelanin. And so neuromelanin is a byproduct of norepinephrine and dopamine synthesis. So usually that signal is bright or rich in places where there's high turnover for production of dopamine and norepinephrine. So things like the locus ceruleus and substantia nigra, and also VTA to an extent. So this is what we're going to use to try and localize the VTA. We'll be doing multi-echo, multi-band functional MRI, whole brain coverage as well with the 7T, and also doing multi-shell diffusion MRI to enable some modeling, microstructural modeling of the data. And I'll go into a bit more detail into each of these over the next slides. But the idea is to use this multimodal high-res data in people with depression and healthy controls to characterize the dopamine system or the VTA network, as well as other networks, of course, that are super important, but just focusing on this one for a start, to ultimately try and link these structural and functional characteristics of the system with these behavioral and clinical and cognitive measures that we're also getting from the participants, either in the scanner or out the scanner. Yeah, so for this, we're going to recruit about 60 people with major depressive disorder and healthy controls as well. So that was the plan. So just to go in a little bit more detail into each of these. So starting with this MT scan, this magnetization transfer contrast scan. So I mentioned the signal will be enriched for large macromolecules like neuromelanin and give us some indication of dopamine synthesis capacity in certain regions. In terms of the workflow, what we do is we acquire the data. We have a magnetom at Mount Sinai and in Oxford. So we actually acquire data with the MT pulses on and off, and then do a division between the two. So in that sense, you can get a contrast enhancement map for that enriched signal related to what we think might be a dopamine signal. We apply some Gaussian mixture modeling to the images. So there we're looking at different signal distributions that we would expect from this type of scan. So one relating to the neuromelanin, one relating to CSF, and one relating to white matter. And you can play around with these different models and the different clusters, the cluster number that you use, but it does a really good job of picking out those voxels that we expect to be rich in dopamine or norepinephrine. And then we do some anatomical masking, but this is the way that we're able to in a data-driven way, localize the VTA and get these voxels that we think belong to the VTA with a little bit of anatomical masking just to exclude some regions outside of the general anatomical area that we expect. And it does quite well. So we do see just on the top row, we do see some within subject stability in this measure. We do see some interesting like sanity check results. For example, the SN it has more dopamine cells, has higher dopamine synthesis capacity compared to the VTA. So we do see that significantly larger signal in the SN compared to the VTA when you're controlling for volume, even though those two signals are highly correlated. So the more dopamine signal you have in the SN, the more you have it in the VTA as well, but they are dissociable. So we see this sort of 0.5-fold enhancement in the SN as we would have expected. And we do see these sex differences. So we do see that females tend to have higher dopamine signal in the VTA compared to males. And this actually recapitulates what we know from animal literature with female mice tend to have exaggerated cell proliferation in the VTA and higher synthesis capacity. So this was just sort of a sanity check, quality check, just to be able to say, can we use this method to localize and measure the VTA? So then in terms of the functional data, so we're using multi-echo functional MRI, the 7T. So this is using this principle of acquiring data at multiple, it's 4 at 3T or 3 at 7T, multiple echo times. And it's using this principle that there is this linear relationship between echo time and bold percent signal change. So that this is kind of shown a bit better here. So you can see at the lower echoes in red, you have sort of a small bold percent signal change, and it linearly increases as the echo time increases. And so when you do this, not only can you acquire data at different points in the T2 star relaxation, which might be important for regional variability in relaxation rates. So that's good. We're getting more data for the optimal T2 star potentially for midbrain and cortex. But secondly, you can use this principle to denoise the data better. So you look for the time series and the signals that show this echo time dependence, and you assume that they're bold-like. And the ones that don't show the echo time dependence, you can assume that they're more noise-like, and you can remove them from the data set. And so you're not doing censoring, scrubbing, things like that, which might be difficult or cause a problem later on. So that's the kind of approach that we use for the functional and the denoising. And I think I showed this earlier, but another comparison, so when you actually apply this method, so this is a functional connectivity map at 3T seeded in the VTA and a bunch of healthy controls. So you do see some connectivity with ventral striatum, a region that we know it's connected to, a little bit of the medial prefrontal cortex. And then when you use 7T, so this is the same statistical threshold, the same people, you get these much broader connectivity maps with much more spatial information throughout the cortex and the striatum. And at 7T, if you actually compare people with depression versus controls, you actually start to see group differences that we didn't see at 3T with the same methods and the same people. You actually start to see this aberrant hyperconnectivity between the VTA and the ventromedial prefrontal cortex or perigenual ACC. And that is actually related to anhedonia. So this is interesting. So why hyperconnectivity in depression? This has actually been shown in preclinical models. So in rats, in rodents, there's a common model called the chronic social defeat stress model. So you put a rat in a cage with an aggressor, a mouse in a cage with an aggressive mouse, and they sort of fight, and you repeat this for 10 days. And sort of the vulnerable mouse develops this pro-depressive phenotype. There's reduced sucrose preference, reduced exploration, reduced social interaction. So there's a number of things that mean that they have this sort of depressive phenotype. And what you see in those rodents is you see VTA hyperactivity. So you see aberrant hyperprojections to the nucleus cumbens, ventral striatum, and some studies shown to the BLA, the basolateral amygdala as well, and the prefrontal cortex. And so this is paradoxical because we know that this connection is important for reward sensitivity, motivation, and learning. And so it's a bit of a strange finding in the literature, but it's been found repeatedly in these preclinical models, and we find it here as well in the human models. And there's the idea that this hyperactivity means that the VTA projections, the dopamine projections to the cortex and the striatum, they're at ceiling level. So the amount of information that they can then convey about rewards in the environment is not feasible. There's no dynamic range there to convey new information. So we're not sure what kind of like cellular changes occur in order to create this hyperconnectivity, but we do have some idea of how it can inhibit function in terms of reward sensitivity and processing, limited dynamic range of the resultant information that can then be portrayed later on. So it was interesting. This was one of the first times people had seen this in depression, and I thank the 7T for this. And so this was a small sample size, but thankfully we recently replicated it in a slightly larger sample size, a separate sample. Again, seeing this hyperconnectivity between the VTA as the seed and VMPFC. If you apply some network graph theory measures to this, you see the same kind of thing. You actually see an increase in influence of the VTA on the whole network, on all the regions that it is supposed to be projecting to. You actually see a higher influence of the VTA on the network in depression compared to controls. Again, associated with this more anticipatory anhedonia. So that was really nice to replicate, because replication is important. And then so that was just all resting state connectivity. So in terms of functional, so we also had participants do a reinforcement learning task in the scanner. So again, we know the VTA is very important for reinforcement learning. So when an unexpected reward appears in the environment, it generates a prediction error. And that the amount of dopamine that is conveyed to the ventral striatum correlates very strongly with the magnitude of that prediction error. And this is a very important signal for learning. So any unexpected reward that you get, you need to learn that stimulus or that context is associated with that increase in reward or that new reward. So this is a task that we use in the scanner just to measure that reward learning, reinforcement learning, and those reward prediction error magnitudes. So it's a two-armed bandit. There's a probabilistic contingency between each stimulus and an outcome. So this one is probabilistically paired with winning, or 20% with nothing, and the reverse for this one. So that's 80% paired with nothing and 20% with winning. So over the course of around 30 trials, people learn which stimulus is associated with a win, but because it's probabilistic, they still get some prediction errors. So sometimes they choose the wrong one and they get a win, or they choose the right one and they get a loss. So you can still model the prediction error, which is super important for learning, and we think is mediated by the VTA and its projections to these target regions. So what we found kind of expectedly in people with depression, they had reduced encoding of these prediction errors in target regions of the VTA. So the BLA, the basolateral amygdala, ventral striatum, as well as the VMPFC, which was interesting. So that was kind of expected. And interestingly, we know from the animal literature there might be some dissociation in terms of VTA projections to the accumbens and these sort of reward-related changes or more anhedonic changes versus the VTA projections to the amygdala and sort of more negative processing, loss, punishment-related processing, and maybe more anxiety-related symptoms. So there is a circuit-specific dissociation in terms of the VTA projections to these different limbic regions and the resulting changes in behavior and symptoms. And we did start to see some distinction there as well. So we saw that this aberrant encoding of prediction error in the ventral striatum was more associated with those like reward-related anhedonic phenotypes, whereas reduced encoding in the BLA, the basolateral amygdala, was more related to those sorts of anxiety-like phenotypes. So I think that was really interesting, and again, important always to look back to the preclinical work to try and inform our models of how we test these things in humans. And just to say, going back to that hyperconnectivity that I mentioned, we did see a relationship between that basal hyperconnectivity of the VTA and reduced encoding of the reward prediction errors in these target regions. So it's not direct evidence, but it kind of fits with our hypothesis that there is a link between this paradoxical aberrant hyperconnectivity and reduced information encoding in the target regions. More work needs to be done in the preclinical literature to see the stages of causality. So just a very brief interim summary so far. We can use magnetization transfer imaging to localize the VTA, I think, in a robust way. We can use resting state multi-echo fMRI to characterize circuitry during rest or during task. And we can use reinforcement learning tasks to probe the actual function of these systems, all in depression, bring it together and try and relate these measures to each other. And I think we might have potentially provided some novel translational evidence of a mechanism by which VTA hyperconnectivity might reduce behavioral function and reward sensitivity in depression. So that's sort of a summary of AIM2 is to continue to do this work and move this forward a bit. I will just say, since I'm here, and for anyone who has been at ISMRM recently, I'm very excited to try echoplanar time resolved imaging as well or alongside multi-echo. So I think this is a very interesting new advancement in how we acquire the functional data, which might be even more important for 7T than 3T. So the idea is that you undersample case base in a very smart, optimized way. So you can actually get many more echo time, data from many more echo times than you could if you were using regular full Cartesian sampling. And so, yeah, so with this method, you can get faster imaging, many more echo times. It's inherently low distortion and low blurring because the echo times that you get are it can be very, very small. So the T2 star decay and the distortion has not had a chance yet to cause these kinds of distortions that we get that are so problematic at 7T. And we're working on applying the multi-echo based denoising approach that I mentioned to this kind of data. So I think this is a very exciting plan for the future that hopefully could overtake some of my other methods that I've been using. But yeah, so part of the work is to test these different methods and with a real specific circuit in mind. Like it makes it easy to just say, like, the VTA network, let's see how well we can do with these different methods just in this one network. Otherwise, it gets a bit too difficult. These methods, they have pros and cons for every different type of application. So for me, I just try to focus on the one thing of interest and test all the methods and compare. Yeah. Okay, so sorry, I haven't mentioned the diffusion yet. So we're also collecting this high-res multi-shell diffusion MRI. The idea here is to be able to fit the NODI model. So this is a geometric model of the diffusion of the water in the tissue. It gives us some indication of the diffusion of water intracellularly and extracellularly as well. So it's not just interesting for white matter, but for gray matter as well. You can get some proxy measures of maybe dendritic complexity and branching. That's the orientation dispersion index that you get from this model. So I think this is a really cool and interesting and important model, so we can look at gray matter complexity as well as white matter fiber bundles and coherence. So I'm showing here, this isn't an image, this is a parameter map of that orientation dispersion measure that I mentioned. And you get really fantastic delineation of striatum, GP, of subcortical regions, a really clear delineation between white matter and gray matter. From a parameter map. It's really fantastic. You can even see potentially some thalamic subnuclei coming out in terms of the different branching and complexity of those subnuclei that you would expect. So not just throughout the brain and the subcortex, but also in the midbrain. This is a slice through the midbrain. You see the SN in white and the bright colors and the red nucleus. Again, this is a parameter map of the dendritic complexity. So you can see it coming out, really being able to pick out these what we think are gray matter subnuclei versus surrounding white matter. And you see in the region that I think is the VTA here, you see much higher complexity measure compared to neighboring white matter. So again, really nice sanity check that this method is working quite well, again at 7T, to be able to characterize these nuclei. And then, yeah, zooming in on the VTA, which is what I'd like to do, we did see some very interesting preliminary relationships between the level of complexity in the VTA and depression symptoms. So here we saw that the more depressed people were, the lower the complexity of the VTA. And again, that seems a bit paradoxical because I said there's hyperconnectivity of the VTA in depression, but it's really interesting to think of a reduced specialization or enhanced specialization of the VTA. So we do see in some animal models atrophy in the VTA, we see degeneration in the VTA and depression models, but this enhanced connectivity, so this enhanced specialization and reduction in variability in the types of processes that the VTA can do. Yeah, so again, this is all sort of proxy correlational evidence, but it does fit with some of the things that we know, and it does explain some of the paradoxes in the preclinical literature. But more to be done there. I'm also, as you can see, interested in testing out different sequences that other much smarter people have developed for the 7T. So I'm interested in working with people in Oxford again, and it's another undersampling of Q space now procedure to acquire diffusion data in a very, very rapid manner. So that data that I just showed you, the multi-shell whole brain diffusion, it takes about 40 minutes to collect at the 7T. And so these guys, Wenchen Wu and others in Oxford, have developed this very rapid acquisition where you can acquire similar data in 6 minutes. So this is something for clinical applications, this is super important. We're losing a bit of resolution, we're losing a bit of specificity. More work needs to be done to compare how well this is actually doing to characterize these nuclei. But for clinical applications, this is really important to have this rapid scan time. So that's something I'm excited to test, but we need to see how well it does compared to our longer sequence in terms of characterizing these small regions. Okay, just to sum up that section again, just to remind you, yeah, that we're using multimodal imaging to try and characterize these networks and eventually relate them to some phenotypes, which I have already started to do, but much more needs to be done in the same sample of people who are deeply phenotyped to relate these measures to the clinical domains of interest. Okay, so third aim is neurofeedback. And I know that some people in this room have been using neurofeedback, so I won't go into too much detail on the methods, but it's this idea that you put people in the scanner, you do some real-time online processing of the data, and you present some real-time neural signal to the participant, and you ask them to modulate it. And so there's different types of signal that you can provide to them. Of course, you might have guessed we're focusing on the VTA. So we're just doing single region neurofeedback. So people are in the scanner, they can watch their VTA activity in real time in the scanner. And we ask them to try and generate a heightened sense of motivation. So as they're doing that, they're trying out various different cognitive strategies, and they can see their VTA signal in terms of like a moving bar, a thermometer. And we ask them to try and get that thermometer to move up using the best self-motivate strategies you can. So through this, they ideally would be able to learn which kind of strategies work best for them in an individualized manner to increase their own VTA activity. So that's sort of the third aim. We did just complete a pilot trial of this at Mount Sinai. So people came in, they did screening and a baseline. They were randomized to either active VTA feedback or sham. Sham is a yoked sham. So it's they're viewing the feedback that somebody else viewed. So it's matched in that sense, but they're not seeing their own brain activity. It's just a single session with about 3 or 4 runs. It's about 30 minutes of training. And then we do post-scan assessments, and we follow them for about a month. Yeah, so we just finished this clinical trial, 61 people, 30 MDD, 30 healthy controls, about 15 per group. So really small sample sizes, but really, again, 7T, sort of a proof of principle feasibility study. Can people with depression even do this? And does it work at the 7T for targeting the VTA? So we did have some interesting and encouraging clinical results. So what we did, because there was so much data that we collected, that depression is incredibly heterogeneous, as I mentioned in the beginning. There's so many different symptom domains that somebody can have. There's so much variability. So we decided to just measure as much as we could in terms of clinical phenotypes and put it all into a factor analysis. So it's a data-driven model rather than looking at each symptom on its own and then trying to correct for multiple comparisons. We just put it all the clinical data into one model. And we found that actually there was one factor that emerged that explained the variance in the clinical change the most. And this represented an overall reduction in a bunch of problematic symptoms. The highest reductions was in this sort of depressed mood and an increase in positive affect. And so what we found was actually that the active group, so that's the neurofeedback group with depression, they showed the greatest factor loadings for this whole reduction in depressed mood and depressive phenotypes factor. So it's a bit all inverted, but they were basically doing better. And the sham MDD group weren't, and the healthies, they kind of were all over the place, but they didn't really have this depressed phenotype to start. So it's a bit difficult to include them all in the same model. We can talk more about that another time, and how to design these things. If you want to include a control group, what are some of the control mood measures? So it's a bit difficult. But just to say is that this factor that represented clinical improvement, it showed that improvement in the active MDD group immediately post-scan, as well as 24 hours after, and it sort of diminished over the course of a few weeks, and it was marginal at 30 days. So we did see, so I would say these are encouraging early signs that this intervention might be useful for some clinical symptoms in depression. And then what was going on in the VTA. So this was sort of a baseline. So blue is the active, so this is a VTA signal, and blue is the active group, and gray is the sham group. So at baseline is sort of all over the place. During training, overall, people in the active group did tend to have higher VTA activity during the training, which was the goal. And then this is a test transfer scan that you do. So you remove the feedback and you ask them how well can they transfer what they learned during the training in a run without feedback. And you see it's diminished a bit, but there is still a little bit of separation. So none of this was statistically significant. So it's a failure. But I mean, yeah, but just to be transparent, to show the graphs, I mean, there are some interesting potential trends. I think the interesting and the key important thing here is not just per se VTA change or per se clinical change, is can we relate people's ability to regulate their VTA to that clinical change? And that is what we saw. So in that final test transfer run, the more people were able to regulate their own VTA, the better they did. And that was true for the overall clinical factor that we found, and also for individual factors. When you just look post hoc at the individual scales, which is also important, we found that negative affect was one of the key symptoms that was most improved. We had some interesting findings with the accumbens and mood, and maybe dissociating some of these different phenotypes, which I can talk more about another time. But yeah, really interesting data set, and yeah, it's very promising, I think. So last little bit, I did have digital phenotyping in the title. So part of my work has been to try and move some of these measures outside of the lab into the real world. So using smartphone apps and stuff to track people in their daily lives longitudinally, which I think is super important. Mood fluctuates dramatically over time, as a bunch of you know. So it's important to try and use some of these measures out in the real world to see how people are doing. So yeah, so for this, we just used a smartphone app and had daily surveys for about a month, people with depression versus control. So you can see, while there is a significant separation in depressed ratings between groups, there's still some variability over time on an individual level in terms of their depression ratings. So this is important to characterize. And even though we do have just one time point for the scan, I think it's important to characterize how people are doing in their daily lives in relation to that scan, in terms of trajectories, variability. There's so much extra information we can get from this data than a single time point questionnaire, which is what we typically use. So for that, we just finished the study, just seeing how feasible this was, how good our measures were in the real world, in about 100 people, half with depression or anxiety, half controls. Just in terms of adherence, we did see quite good adherence over 30 days, about the same whether you're depressed or not, and it didn't drop off over time, which we kind of expected. So this is just one small sample of 100 people, but there are some measures of engagement that you can do with participants to make sure that they're sticking to it and they're adhering. So in this study, it was quite good, the adherence. And so the first question was, do our real world sort of digital single item measures of depression, anxiety, and related phenotypes, do they relate to the sort of gold standard in lab, like 40 item scales that we usually use? We did see quite strong relationships between anxiety, senses of distress, and anhedonic symptoms between the full items and the daily items. And we did measure some passive data as well, like steps, screen time, that kind of thing, we did see an expected relationship between physical activity and depression. So the more active you are, the less depressed you are. There's an interesting lag. So it's usually the effects are more pronounced on mood a few days after you've done the physical activity, which is interesting. Again, it's a small sample size, so it's preliminary, and there are other groups doing this in much larger data sets. But just to show that these kinds of tools are available, and they work on these sort of smaller data sets as well to supplement the clinical trials that we run and the cross-sectional imaging scans that we run. I think it's important to use this kind of data. Something I've been getting a bit interested in as well is measuring people's faces in the real world, and as they're using their phone or they're interacting with a cognitive task on their phone, for example, or a questionnaire. So there's some interesting work by a group, I think in Dartmouth in the US. They developed this app called the mood capture app. So they had just had people with depression rate their symptoms every day, similar to us, for 30 days. They were also just recording their face while they did it. Everyone knew that their face was being recorded, but they were able to pick up expression information from their face that could predict their mood. And these were people who had quite significant variability in their mood. So sometimes they dip in, dip out of the depressive state, and the facial expression could pick up on that and distinguish between what kind of state they were in. It's super creepy, but I think it's really interesting and important for these sorts of objective, non-invasive ways in which we can help track people's symptoms in the real world, help the individual know what kind of state they might be going into based on how maybe their facial expressions are changing. And I think there's a lot of commercial and national security teams that are doing this already. And I think in academia and we need to kind of catch up with some of these tools that are already being developed and use them for the good, and to try and help clinicians and patients understand each other and themselves. Yeah, sometimes the face can tell us, as clinicians know, the face can tell us a lot more than what a person can verbally say, or the insight that they might have into their current cognitive emotional state. Yeah, I think I might be running out of time. I'm not sure the exact time. Okay. So just to say that for this, I'm planning to do this in the studies that I described, using this very cool AI facial expression and voice sentiment expression model called Hume AI. It was trained on very diverse data set, facial data sets. It was also very well trained and tagged by culturally very diverse individuals, rating what each type of facial expression meant, because there's a lot of cultural variability in how we interpret what a facial expression means. I think it's a very, very robust algorithm, and I think it's very cool, not just for face, but for voice sentiment. It performs very, very well. So the plan is to have people, as well as using the app to answer surveys every day, they'll just do a 2-minute video diary every day, just asking them how's their day going. It can be prospective, it can be retrospective, how their day's been, how their day's going, whatever they want. 2 minutes, recording their face, we get their voice, we get the content, we get their face, and we're hoping from this we can try and determine how people's moods might change. Is there going to be a change point coming up? There's a bunch of things that we can do with it. Just to give you an idea of some of this facial expression data. So we have some recordings of clinical interviews with patients. So this is the SCID for DSM-5. And so this time series is the estimated % joy expression in the yellow that the person has during the course of the interview. And in blue, lower, it's the % sadness expression of their face. And when you just summarize the data, you see a huge dominance of sadness in their expression over joy, which you would expect. So many different analyses you can do with this data. You can look at the composition between these different types of expressions. You can look at changes over time. You can look at network interactions and influence, and how does sadness influence joy in terms of temporal changes. So the same kind of analysis that we perform with our functional time series data, you can apply to this multifaceted data as well. Just to compare, that was just one person, a young male with depression. Just to compare to a control participant, you see sort of the flip. So joy dominates and sadness is underrepresented. But interestingly, that person sort of showed a bit more of a healthy mix between a range of different facial expressions rather than just being stuck in one sad phenotype, a sad facial expression. So you can start to think how many interesting models we can fit to the data to understand it. Okay, summary. I think I've said that it's important to deconstruct and measure anhedonia and depression and in a range of clinical participants as well. We have an interest in measuring intrinsic motivation, and we have a couple tools to try and do that. It's important to test out a bunch of different high-res multimodal neuroimaging methods to study different aspects of the brain, structure and function, microstructure, and apply those to how we understand behavior and clinical phenotypes. And potentially neurofeedback might be a novel probe of the systems that we're interested in, not just a clinical intervention, but also a probe of changing network functional interactions and maybe seeing how that relates to behavior thereafter. So that's it. Please be in touch with any questions. Love to chat and collaborate. And thank you.